Okay, thank you for that. Okay, so we are going to be looking at the inner ear mostly today. And there are only two structures labeled in there that are relevant for today, the semicircular canals and the vestibular nerve. We are going to be more looking at more structures. And hopefully by the end of the class today, you're going to be able to recognize and describe what's the role of each one of the structures labeled in there. And it looks overwhelming, but let's take one step at a time. We already looked at part of this last, let me move this here, in last class. The external auditory canal receives and amplifies the sound that impacts on the tympanic membrane. The tympanic membrane then translates that wave of sound into the malleus, the incus, and the stapes. And the stapes sits on top of the oval window. These three small bones are connected also with the stapedius muscle and the tensor tympanal muscle that prevents from really loud sounds to affect this structure. So I mentioned the stapes sits on the oval window and translates that movement into the liquid inside the cochlea. Inside the cochlea, we described last time the scala vestibuli and the scala tympani, both of them filled with perilymph, and the cochlear duct or the scala media that is filled with endolymph. Okay? And once the wave traveled inside the cochlea and went through the basal membrane, it exits through the round window. The pressure exits through the round window. And I mentioned that it deflects or it travels through the basilar membrane, and basilar membrane detects higher frequencies towards the base of the cochlea and in lower frequencies in the apex. And then that information is transported into higher cortical areas through the cochlear part of the cranial nerve number 8. So all of this is a brief summary of what we saw last week. So out of this schematic, half of them we already know what they are. Let's take a look at the rest. And what we are going to be talking about today is the vestibular labyrinth. And it's composed of 5 sensory structures. 3 of them are the semicircular canals: the superior, the posterior, and the horizontal semicircular canals. And 2 of them are the otolith organs, the utricle and the saccule. There are 2 definitions also that I want to introduce now. The bony labyrinth is the rigid outer wall of the inner ear. And the membranous labyrinth, which is a series of tubes that are filled with fluid that we are going to describe in detail today. And those tubes are the ones that contain the receptor cells. Here is another diagram of the inner ear. You already know this part. This is the cochlea. This is the auditory part of the cranial nerve number 8. In here are the labyrinth structures. Here we have the 3 semicircular canals. At the base of each one of them is the ampulla or the ampullae in the 3 of them. At the base of this is the utricle and the saccule. And as you can see, the cochlea and the semicircular canals, the utricle and the saccule, are connected by the canals reunions. And I'm going to be talking about that a little bit more later on. There are neurons in the Scarpa ganglion that extends into the sensory neurons in the semicircular canals and the otolith organs, and axons that project into the central nervous system. Again, another schematic. I'm using each one of them to reintroduce some of the subjects and also to introduce new concepts. Again, the cochlea, you already know. The otolith organ, the utricle, and the saccule are located in here. At the base of each one of the semicircular canals is the ampulla, And in this particular schematic, through transparency, you can't see this from the outside, you can see the cupola. And I'm going to be talking about that later on. What I want to the concept that I want to introduce in here are basically two. One of them is that the semicircular canals receive different names. And I try to modify all the images that I have in here, but you're going to find schematics or drawings in books that have all three or all four names. So that's why I'm introducing them, all of them here. I'm going to stick to superior, horizontal, and posterior through the length of the talk. Okay? And there's one more thing that I want to draw your attention. Basically, in this drawing, there are two colors. One of them is yellow-ish, like this one, and the other one is gray. And it's going to become important later on. I'm going to bring it back later on. So the cochlea and the vestibular system are in the petrous portion of the temporal lobe. And here is a representation of where they are, both on a lateral view and on a top view. If we look into detail into the position of the horizontal canal, it's almost at 30 degrees. The orientation is 30 degrees compared to the horizontal line. And it's going to become important later on to understand how it works. What about the other two? The posterior and the superior, located here and here. If you start paying attention at the orientation of them, you're going to realize that this one, the posterior, is in the same plane than the superior on the other side. And the same happens with the superior and the posterior on the right and the left. So what I'm trying to, the concept that I'm trying to introduce is that the semicircular canals are organized in complementary functional pairs. The right superior works together with the left posterior. Both horizontals work together, and the right posterior works together with the left superior. Okay? Just because they are in the same plane of action. Okay? They're going to be detecting the same type of movement. That is the position of the semicircular canals. The saccule and the utricle are located at the base of the semicircular canals. Okay? I know I'm repeating these concepts over and over, but it's so you understand later on how all of these structures work. Before we move on, I want to introduce basically three definitions. One of them is that any object has 6 degrees of freedom. One of them, or three of them, sorry, are translational. One of them is in the horizontal plane, one of them is in the Z plane, and the other one is in the Y plane. Okay? And they have 3 rotational movements, which is along those planes. It could be on the X, it could be on the Z, or it could be on the Y. Okay? The other 2 definitions that I want to introduce is linear acceleration. It's the rate of change of velocity without change in direction. A good example would be when an aircraft is increasing speed without changing the pathway. Okay? Like on a runway before taking off. That's a good example of linear acceleration. Angular acceleration or rotational acceleration is a quantitative expression of a change in angular velocity that a spinning object undergoes per unit time. That is a little bit more complicated, but imagine basically doing this with your heads. Okay? That describes an angular velocity. Okay? It's it's not exactly, like the same effect on the swinging chairs on a theme park. Okay? When they're spinning down, you have an angular velocity. So, the saccule is oriented in such a way that it's going to detect linear acceleration in the vertical plane. So it's going to detect head tilts and vertical displacements. Okay? So it's located like this. So any movement up and down or tilts that are linear acceleration is going to be detected by the saccule. The utricle is located like this. So it's going to detect linear acceleration in the horizontal plane and tilts of the head. So far, so good. It's going to get more complicated. The semicircular canals detect angular rotation. And they detect the acceleration of the angular rotation. Okay? So the same example that I used before, turning the head like this, really fast, or spinning as the first images that I have on the slide, like an ice skater spinning, that is influencing, basically, the semicircular canals. Okay? So, let's go back to another schematic, but now instead of looking at the outside of the inner ear, let's look at the inside. This is the part that you already know. This is the cochlea. In the cochlea, we have at the apex the helicotrema, where the scala vestibula and the scala tympani connects. And in the middle, we have the cochlear duct or the scala media. The scala tympani and the scala vestibula are filled with, louder? Perilymph, perfect. And the cochlear duct? Endolymphs. So here what colors are showing on the screen? Almost no color. Okay, so on this one, the light gray in here represents the endolymph. The whatever color is that represents the perilymph. Okay? Here we have the stapes, and here we have the round window. As you can see, the saccule is connected with the inside of the saccule is connected with the cochlear duct through, I mentioned before, the canal ray unions. Now we are looking inside, so the connection is made through the ductus ray unions. And here is the saccule, here is the utricle, here are the semicircular canals. All of them painted with the same color, meaning all of them are filled with the same content, which is endolymph. And all of them surrounded by perilymph. There is also an endolymphatic sac and an endolymphatic duct and a perilymphatic duct that regulates the contents of the endolymph and perilymph and also somehow involved in immune responses. So how is this possible? How is it possible that the cochlea and the saccule are connected? In order to understand that, we have to go back in time and look at the embryological development of the inner ear. Here we have the otic placode. The otic placode starts invaginating and it forms the otic pit. And then it separates from the ectoderm that is on the surface, and it forms the otic vesicle. I showed you briefly this, and I said, like, when we look at the vestibular system, we are going to keep on going with this. Last time, we focused mostly on the development of the first pharyngeal punch and the first pharyngeal cleft. So, how this otic vesicle develops? It starts elongating and forming the endolymphatic sac and duct, and then it forms two structures, the saccular portion and the utricular portion. Let's look a little bit more at how the saccular portion develops. It starts elongating and twisting, and it forms a cochlea. But it remains connected with the saccule through the ductus reunions that I just showed you on a fully developed inner ear. This is what I just explained. What happens with the utricular portion? It forms a utricle, but also it starts forming the semicircular canals. The walls in the middle get attached to each other, then they disappear, they get degraded, and it forms the semicircular canals. And that is how, in the end, we have all these canals, the utricle, the saccule, and the cochlea, all connected and filled with the same fluid. Okay? So, we talk about endolymph and we talk about perilymph. And I already explained all of this to you last time. The endolymph is formed from the stria vascularis and it has a high content of potassium, and the perilymph has a normal extracellular content of potassium, which is 7 millimolar. Okay? And you're an expert on this because I explained this before, but I'm going to go through this one more time. So, hair cells are in contact with the endolymph. Okay? And in resting, when there is no sign, and I'm using sign. When there is no sound, and I'm using sound because that's the way I explained it before for the auditory system, there is potassium flowing in the cell. When the cell moves towards the higher, or the stereocilia move to the higher stereocilium, then those channels open even more, and there is an increase of influx of potassium. And if they move towards the shorter one, those potassium channels close, and there is a decrease in the entrance of potassium. The entering of potassium induces a depolarization of the cell that triggers voltage-gated calcium channels, which in turn produces an influx of calcium, and that influx of calcium releases vesicles with excitatory neurotransmitter that impacts on the afferent neurons. And the fact that at resting these cells are firing, it gives us the possibility of increasing the firing when the cell depolarizes, like shown in here, or decreasing the firing when the cell hyperpolarizes. And that is information that the brain senses in order to understand what the sound is. Why I'm going back to sound and hair cells in the vestibular system? Because the structures that are going to detect motion are very similar to the ones that we found in the inner ear, in the cochlea, sorry. Everything is the inner ear. Here is the structure. This is a cross-sectional sagittal view where you can see the hair cells with the stereocilia and the kinocilium, all of them oriented in one way, and all of them innervated by afferent cells. Surrounding them, we have supporting cells. Here we have a top view, so now we are going to be looking at this image from the top. Here is how they are oriented. And the arrows, can you hear me? And the arrows are pointing into the direction of the depolarization. Okay? So this is the way that hair cells are organized inside the vestibular system. So, as I said, the vestibular system contains hair cells that provide the basis of vestibular function, and these hair cells transduce displacement into receptor potentials. And I'm going to explain that. I already mentioned the semicircular canals detect angular rotation acceleration. How do they do that? Let's take a look at the ampulla that is at the base of each one of these semicircular canals. Inside the ampulla, what we are going to find is a cupula. That is a gelatin-based structure. It's a little bit thicker than the endolymphs that is all around. Inside that gelatin-like structure are the stereocilia that come from the hair cells that are located in the crista. And these hair cells are innervated by afferent axons. Here, what I'm showing is the orientation of all these stereocilia. All of them are oriented towards this side. Okay? The flow or the movement of the endolymph inside a semicircular canal can be or is bidirectional, can move both ways, and can persist after movement of the head stops. And I'm going to use an example. It's not the best example, but it's going to do the trick. Hopefully, all of you at some point try to move a bucket containing water. And when you move it, the first thing that it happens is that the water moves exactly in the opposite direction, correct? And it forms a wave on the opposite direction. It's not entirely true that it moves in the opposite direction. What is happening is that you're moving the bucket, and the liquid lags behind. Okay? So when you move the bucket, it takes time for the liquid to move. Okay? And when you stop, basically, you splash the water all over you because the water now keeps momentum, right? And it moves towards where the movement was. So that's why I'm trying to say that the endolines can move both ways, and it still moves after the movement stops. Why I'm telling you all of this? Because when you move your head, all the bony structures, all the bony labyrinth are going to move with you, okay? But the endolymph that is inside the semicircular canals is going to lag behind. So basically, let's make it with a horizontal canal. If the head turns like this, the liquid inside is going to lag behind and push the hair cells exactly in the opposite direction. The head is moving, is having an angular acceleration this way. The endolymph flow lags behind and pushes the hair cells in the opposite direction. So far, so good? So I already explained to you how hair cells work. So basically what we just did in here is we transduced movement into an electrical signal. The electrical signals I mentioned before is the language that the nervous system understands. Okay? Because the hair cells, by moving this way, it induces the depolarization of the hair cells. Everybody understands that? So, how does it work? That is in detail how it works inside one ampulla on one semicircular canal. Let's use as an example the twisting of the head in the horizontal plane. Okay? So doing something like this. Okay? The head moves into this direction. The hair cells, the axis of the hair cells is like that. So the fluid is going to lag behind and move in this way, depolarizing the cells, which is going to increase the firing rate on the side where the head was moving towards. So the head is moving towards the left semicircular, horizontal semicircular canal is going to increase the firing rate. What happens with the right one? The right one, the bony structure, is going to move exactly the same way, and the endolymph is going to respond exactly the same way. Okay? But the hair cells are oriented in the same way as they are oriented here, facing anterior. So now the movement of the endolymph is going to hyperpolarize the cells, decreasing the firing rate. So the brain can sense those differences and understand that you're moving your head towards the left. I explained that for the horizontal canals because it's probably the simplest one. But this push-pull arrangement operates for all three pairs of canals. And I mentioned before how they were aligned in the same plane, the different canals in the right and the left. The pair whose activity is modulated is in the plane of the rotation. Again, I use the horizontal as an example because it's the simplest one to explain. The member of the pair on the side toward which the head is moving shows increasing activity. In my example, the head was moving towards the left semicircular, horizontal semicircular canal increased the firing rate. And the member of the pair on the side away from where the head is moving shows a decrease in activity, hyperpolarization. Okay? What about the otoliths organs? The saccule, I mentioned this before, is oriented to detect linear acceleration and head tilts in the vertical plane, while the utricle also detects linear acceleration, but on the horizontal plane. How do they work? They are called otoliths organs, basically because from the Greek, otolith means ear stones. And when we look at the structure, at the sensitive part of the structure of the otoliths organs, we're going to see that there are hair cells with stereocilia, and one kinocilium, afferent fibers, the supporting cells. All the stereocilium are embedded on a gelatinous layer similar to the cupula on the semicircular canals, but there are otoconia on top of that gelatin. Those otoconia basically are like small sand structures. Here is scanning electron micrographs showing a kinocilium over there, stereocilia in here, the gelatin-like structure, and the otoconia. And here is a higher magnification of the otoconia. Okay? So these otoconia sit on top of that gelatin, putting pressure on top of the hair cells. And if you look into detail, you're going to see that half of the hair cells are oriented one way. Here is the kinocilium towards the middle line, and here is the kinocilium on this one and the rest of them. All of them are oriented towards the striola in the midline. And that is how the utricle is, the sensitive part of the utricle looks like. They are oriented towards the striola. In the sacculus, they are oriented away. Basically, what this tells you is that when you make a movement, half of either the utricle or the sacculus is going to be hyperpolarized, while the other half is going to be depolarized. Okay? Again, that is important for the brain to understand what is going on, sounds weird, inside your head. You make a tilt with your head. So basically, that is what's going to happen. And what is happening at the same time is that the otoconia are going to be attracted by gravity. So they're going to put pressure on top of the hair cells. And that pressure is what is going to make a deflection of the stereocilia, making them to hyperpolarize or depolarize. There is one little thing, it's not a problem, but a little thing that the utricle and the sacculus have, and is that they can detect linear acceleration, but they cannot make a distinction between a head that is tilted or whether there is, excuse me, whether there is acceleration in the horizontal plane, for example, excuse me. There is no way for either the utricle or the saccule to differentiate one from the other. So how do we know that we have the head tilted or that we are just walking? When explaining all of this, I'm trying to oversimplify how it works. Basically, none of them work on its own. Head and body motions are interpreted by integrating input from the otoliths organs, the semicircular canals, the visual system, and the somatosensory system. All of them help in understanding what's the position of the body, what's the position of the head, how is the head moving at any given time point. We're trying to separate how each one of them work, but remember, none of them work on its own. Okay? Although both of them, the semicircular canals and the otolithic organs, have hair cells and depolarization and hyperpolarization is the same, they don't respond equally to the stimulus. On the left, we have the semicircular canals. When there is an acceleration, there is a phasic response of the hair cells. Why is that? Because when we are accelerating, the liquid lags behind for a while. But if we reach one particular speed, the endolymph inside is going to catch up. And there's not going to be more stimulation because of that. Okay? And that was why if the velocity is constant, then there is a decrease in the firing of the semicircular canals. Once we stop, again, the endolymph lags behind, keeps on moving, but now it's going to produce a hyperpolarization of the cells and then reach the normal status. So semicircular canals respond in a phasic way. What about the otoliths organs? We start tilting our head, so we have a response. And we keep the head tilted. And the response, while the head is constantly tilted, the response still exists. Why is that? Because I mentioned it briefly, but what is affecting the response on the otolithic organs is gravity attracting the otoconia. And gravity is in there all the time. So if we have our head tilted, gravity is going to keep on pushing the otoconia, and if they were depolarizing the cells, they are going to keep depolarizing the cells until we move the head back and the response disappears. So the response of the otoliths organs is tonic compared to the response of the semicircular canals. Everybody understands that? Okay. Here cells from the labyrinth are innervated by bipolar cells of the Scarpa ganglion. I mentioned the Scarpa ganglion before. And the neurons of the Scarpa ganglion project centrally via the 8th nerve of the to the vestibular nuclei on the floor of the fourth ventricle. And they innervate the inferior, lateral, medial, and superior vestibular nuclei. And again, I'm reinforcing this, central vestibular processing is multisensorial. The vestibular nuclei receive input from the cerebellum, the visual, and the somatory sensory systems. Okay? You already know this, you saw this with Dr. Harrison. Here are the vestibular nuclei. And I want to point out how long it extends in between the medulla and the pons. Okay? So if we see, if we look at cross sections at different levels, we are going to see the vestibular nuclei at different levels. Again, this is pointing exactly the same thing. Here are the vestibular nuclei, superior, lateral, medial, and inferior. Okay? They are spread along a long way through the medulla and the pons. And I'm going to show you one section that is at this level, at the level of the cochlear nuclei. Again, I'm stealing this from Dr. Harrison's presentation. Here is the ventral cochlear nucleus, here is the dorsal, here are the vestibular nuclei. Okay? So I mentioned, the vestibular ganglion cells project into the vestibular nuclei. It's not the only place they project to. They also project to the cerebellum, to the vestibulocerebellum, to the flocculonodular lobe. And you already know this from anatomy, but this is a ventral view of the brain, and here is where the flocculonodular lobe is located. Okay? So we have information that goes from the vestibular ganglion to the cerebellum, but also to the vestibular nuclei. And from the vestibular nuclei, through the medial longitudinal fasciculus, there are ascending components and descending components that go into the spinal cord. The ascending components go into the abducent nucleus, the trochlear nucleus, and the ocular motor nucleus, as well as the thalamus. Why it goes to so many different places? Because the central projections participate in two major processes. One is maintaining equilibrium and gaze and coordinate movements of the head with movements of the eyes, and also for maintaining posture. In order to do that, it uses three different reflexes. The first one is the vestibulo-ocular reflex, which excites, inhibits opposite nuclei controlling eye muscles to compensate for head movements. I'm going back to this example. We have our eyes fixed on something, but we move the head towards the left. How do we keep our eyes fixed on that structure? Turning of the head to the left, increase firing of the semicircular canals, hair cells on the left. Here is the left side of this image. Okay? Here are the scalpa ganglion cells. They project into the medial vestibular nucleus, nothing new so far. From there are two projections. One excitatory that crosses the midline and goes into the abducens nucleus, and one inhibitory that remains ipsilateral. From the abducens nucleus, the projections go to the lateral rectus or to the medial rectus going through the oculomotor nucleus. In order to get to the oculomotor nucleus, axons have to cross the midline again. Okay? This is the excitatory path, which excites the lateral rectus on the right eye, and through this pathway, the medial rectus on the left eye. Here is the inhibitory pathway. And it follows exactly the same path, but now there's an inhibition, which ends in an inhibition of the medial rectus on the right and the lateral rectus of the left. All of this, basically, to turn your head and keep your eyes in the same position by contracting the muscles on the right and relaxing the muscles on the left. Okay? There are two more reflexes. One is the vestibulocervical reflex. Basically what it does is represented in here. Basically what it does is excites neck muscles via the medial longitudinal fasciculus. Here is the medial longitudinal fascicular. So basically we can move our head, the muscle is in our neck, in order to move our head towards where we want to move it. And the other one is the vestibulospinal reflex that excites ipsilateral limb or trunk extensors to oppose body tilt. So basically, at the same time that we are moving our head, we have to compensate for that movement with the upper part of the torso. So all of them are working together when we are moving our head. And all of them imply the vestibular system. I mentioned that from the vestibular nuclear there are projections that go into the thalamus. And from there, from the thalamus, they go into the vestibular cortex. It's a rather diffuse area, but mostly is the region near where facial representations are located and in the posterior parietal cortex. But in these areas, there are also a lot of integration from somatosensory and visual, and bringing this over and over. When we move, basically we have to compensate and understand what is going on. And that the information that comes from our body as well as the visual system help in maintaining posture and gaze. There are also descending fibers that basically arise from the nucleus abducens. And it seems that they increase excitation of the hair cells and may modulate the dynamic range to match expected or anticipated acceleration. There's some evidence about on this, but it's not definitive. So, we're going to move now into and talk a little bit about disorders of the vestibular system. You're going to have a class about disorders of the vestibular system later on. So I'm going to introduce some terms so it's easier for you to understand the class later on. I'm going to define first a physiological nystagmus, and it's the alternating slow and rapid eye movement that is part of the vestibular ocular reflex. So, basically, what is shown in here is that when the head turns, there is an increased firing on the side where the head is turning to and a decreasing firing on the on the side where the head is moving away from. That difference in the firing rate is sensed by the brain, and the response, the ocular motor response, is a slow movement towards the side of the where the head is moving, with a fast saccade moving in the opposite direction. If one of the canals is not working properly, or is, in this example, is not working at all, the brain is going to sense this difference as the head is moving, even if it's not moving. But there is, for the brain, a difference in the signaling that I'm getting from the right and from the left. That means that my head is turning. And that is pathological. It's a pathological nystagmus. And it's the alternating slow and rapid movement indicative of vestibular pathology when there is no head movement. Okay? There are some tests that you can do in order to test the integrity of the vestibular system. And one of them is called the caloric nystagmus. Basically, in a normal person, if you irrigate one ear with cold water, the person is going to have a slow movement of the eyes towards where the cold water is, with a fast saccade in the opposite direction. But if you put warm water, the slow movement is going to be in the opposite direction, with a fast saccade towards where the hot water is. Basically, what you're doing with the cold and the hot water is increasing the fluidity of the endolymphatic liquid inside the semicircular canals. And by increasing or decreasing the density, basically you're increasing or decreasing the firing rate of the semicircular canals, and that is sensed by the brain as moving of the head, even though the head is not moving. In unconscious patients, if there is a lesion, if the brain stem is intact and you apply cold water, you're going to have the normal slow movement of the eyes towards the side where the cold water is, without the fast saccade, because the patient is unconscious. If you have a lesion on the medial longitudinal fasciculus, only the eye closer to the ear that you're testing is going to move. And if the lesion is in the low brainstem, there is not going to be any response in either one of the eyes. Same thing applies to the warm water, but exactly in the opposite direction. Okay? Vertigo. Vertigo is the sensation of the room spinning, and is often caused by an inner ear problem. One of the causes could be a benign paroxysmal position vertigo, or BPPV. And it occurs when the otoconia clamps in the semicircular canals in the inner ear. Remember, the otoconia are in the saccule and the utricle, not in the semicircular canals. But sometimes they can detach and they can get into the semicircular canals. Using the Dix-Hallpike test, you can detect whether a patient has BPPV. And sometimes, using an Epley maneuver, you can remove the otoconia that got stuck into the semicircular canal. Another cause for vertigo is Meniere's disease. I'm going to talk about that in a minute. Or vestibular neuritis, that is usually related to an infection. And the infection, what causes is an inflammation of the inner ear around the nerves that are related to the sense of balance, but there is no alteration on hearing. And the treatment depends on what it caused the vertigo. In many cases, the vertigo goes away without any treatment. Meniere's disease. I mentioned Meniere's disease at the end of the auditory class, and I said that we're going to talk a little bit more about that in the vestibular class. Why? Because it's a disorder of the inner ear that causes severe dizziness, vertigo, ringing of the ear, tinnitus, I mentioned that in the auditory class, and hearing loss. And usually Meniere's disease only affects one ear. The symptoms are caused because the liquids inside the inner ear build up, and the pathology suggests that the endolymph builds up in the labyrinth and interferes with the normal balance and hearing signals. Remember, the endolymph in the cochlea and the endolymph in the membranous labyrinth are all of them connected. What causes Meniere's disease? There are many theories, but no definitive answer of like exactly what is the cause. Could be different reasons why it happens. What's the treatment? One treatment is medications. Prescription drugs can help relieve the dizziness and shorten the attacks. Because there is a building up of the liquid of the endolymph inside the semicircular canals and the cochlea, salt restrictions and diuretic help a lot. And also injections. You can inject an antibiotic. The injection is done in the middle ear. It helps control vertigo, but increases a lot the risk of hearing loss because the gentamicin can damage the hair cells. Or the other thing is injecting corticosteroids, which usually helps reducing the dizziness with no risk for hearing loss. Motion sickness. Motion sickness occurs basically when the data provided by the vestibular system is not consistent with the data provided by the visual or somatory sensory systems. Basically, is what happens when you're in a boat. You're inside the boat is moving, but your eyes don't sense that anything is moving. But the vestibular system says, like, yes, something is moving. And that's how you end up throwing up inside a boat. Okay? Well, not all of you. I do. Luckily, the sense of balance is plastic, and we can like adapt in 72 hours to be in a boat in the sea. If we are long enough in the sea, we can have the same problem when we get back into land. The vestibular system adapted to movement, but the eyes are saying like nothing is moving, and it could take some people some time to readapt after being a long time in a boat to be in land again. Finally, the question you've always wanted to ask but were afraid to ask. Why does the room spin when you lie down after a tough evening? And I'm not saying that this happened to any of you. Probably a friend, you saw that in a movie. Why after drinking a lot of alcohol, everything spins? I mentioned before that the endolymph has one source, the striatum vascularis, of blood. The cupula has its own source of blood. So alcohol dilutes the blood, and that goes into the cupula faster than it goes into the endolymph. So basically, the cupula, the gelatin-like structure inside the semicircular canals, is lighter. So when you basically rest, because it's lighter, has fewer density, tends to float. But at the same time, deflects the hair cells in there. So when you put your head down, the brain says, oh, the room is spinning. No, it's not. And you feel terrible. What happens after? Well, the cupula, as the blood is drawn from the, the alcohol is blown through, from the blood, it changes. But the endolymph that is being produced now has a higher content of alcohol. So now the cupula has a higher density, tends to move towards or to fall, like shown in here, and again you feel terrible. So basically that is why the room spins after you, not you. Somebody has a lot of alcohol. So I mentioned this at the beginning, that we were going to hopefully be able to understand what each one of these structures do at the end of the class. We talk about all the ones that are shown in here. We talk about the membranous labyrinth and the bony labyrinth. The bony structure, the bony part is where the inner ear is located. We mentioned that they are filled with endolymph and surrounded by perilymph. We talk about the ductus reunions that connect the otoliths with the cochlea. We mentioned the endolymphatic duct and how it regulates the contents of endolymph. We talk about all three semicircular canals and how at the base of each one of them there is an ampulla. We talk about the utricle and the saccule, and the vestibular or scarpa ganglion that projects axons into the hair cells and then axons into the central nervous system through the vestibular part of the cranial nerve. Okay, so we are going to make a break now, and we're going to talk about the taste and olfactory system after this. let's start with the second part. In the second part of the talk, we are going to talk, or the second part of the class, we are going to talk about chemosensation, also known as taste and olfaction. And we are going to start with, it's recording, right? No. Okay. So, gustatory system. There are basically 5 established basic categories: sweetness, sourness, saltiness, bitterness, and umami. There are other categories that not everybody agrees upon. Some people say that they can taste fat, astringent, the CO2 that is on a soda, or pungent tastes. Though there's a lot of argument about this one, all of these ones being true or real. Where do we sense different tastes? Where do we sense bitter, sweet? For years and years, and there are many, many, many books that have this diagram, that bitter is sensed in the back of the tongue, sweet in the tip of the tongue. That is not true. That comes, like if you go into history, from an over-interpretation of something that somebody said once and it stuck with us for years. But it's not true. Okay? What we have in the tongue are different types of papilla. Circumvallate papilla are in the back, foliate towards the side, and fungiform papilla in the tip, on the most anterior part of the tongue. The circumvallate are innervated by the cranial nerve number 9, the glossopharyngeal nerve, while the other 2, the foliate and the fungiform, are innervated by the chorda tympani, the 7th cranial nerve. Here is a structure of a circumvallate papilla, a foliate, and a fungiform. And each one of them contain taste buds. One taste bud, here is a representation, a real section of a circumvallate papilla. Each taste bud looks like this. It contains supporting cells, taste cells, and basal stem cells. And also, there are afferent connections that make a synaptic contact on the taste cells. And here is a scanning electron, a transmission electron micrograph of this representation. In the pore, in the taste pore, is where we are going to detect different molecules. Each single taste bud contains between 50 and 100 taste cells, and all 5 taste sensations are represented in any given taste bud. Okay? That's why we can taste all of them across the tongue. There are no particular areas where we are going to taste sweet or bitter. There are basically 2 types of receptors. One for taste. One that are ion channels, and those are the ones involved in detecting salt and sour. And then we have the GPCR, G-couple protein receptors, and those are the ones that detect bitter, sweet, and umami. How do a taste cell works? In here we have a representation of an ion channel that might detect salt or sour. Here we have a representation of a G-couple, a GPCR, G-couple protein receptor that can detect sweet, bitter, or umami. Remember, these are in different cells. This is just a schematic showing a general mechanism for all of them. Okay, but imagine that I can make 5 different cells, each one for one of the tastes. So either salt or acid binds to the ion receptor, it opens and ions flow into the cell, or they bind to the GPCR receptor, which activates a G protein that in turn activates second messengers that are going to induce the release of calcium from the endoplasmic reticulum, or that they are going to induce a depolarization of the cell. That depolarization increases the concentration of, the input of sodium and calcium, and that entry of calcium in either one of those pathways release induces the release of neurotransmitter, serotonin in particular, that is going to induce a depolarization into the afferent neuron. One thing that is important to mention in here is that taste receptors and olfactory sensory neurons, as I'm going to show you in a few minutes, are the only sensory neurons capable of generating action potentials. Okay? When you look at the visual system, or when we talk about the vestibular or the auditory system, we never talk about action potentials on the sensory cells per se. Taste cells and olfactory sensory neurons can have action potentials. And that is what releases the neurotransmitter at this synapse with the afferent fibers. So, I mentioned this before. The facial nerve, the choroid tympani branch, innervates the most anterior part of the tongue. The glossopharyngeal innervates the most posterior. There are also taste buds that are found in the upper, in the part of the GI tract, in the esophagus. Lately, it has been discovered that there are also taste receptors throughout the GI tract. These innervations go to the nucleus of the solitary tract, which is a gustatory area in the brainstem. And again, as I did before, I'm going to steal some slides from Dr. Harrison. She showed you this example. Now I'm pointing out to where the nucleus of the solitary tract is located. And here, again, stealing from Dr. Harrison, here is the nucleus of the solitary tract that she showed you a few classes ago. So, another representation of the same thing, cranial nerves 7, 9, and 10 going into the nucleus of the solitary tract, the gustatory area in the brainstem. From there, there are projections that go into the thalamus, and from there it goes into the frontal taste cortex. The complete pathway is the one shown in here. Okay? It involves the nucleus of the solitary tract, as I just mentioned. Then it goes into the thalamus. From there, it goes into the insular, here it is, and the frontal cortices, and from there into the amygdala. And part of this pathway has to do with the reward part of eating. Basically, the satisfaction that produces into us to eat some particular things, and also into society when we need to stop eating. When we talk about the auditory system, we talk about the tonotopic map and how the sound that is detected in the cochlea at different levels have a representation in the cortex. What happens with the gustatory system? There were some experiments done in mice in particular that told us that the afferent fibers are the ones encoding for what we taste, not necessarily the taste cells per se. How did I know that? Well, here's an example. This is a wild-type control, an animal that is just drinking water. If in the water we put a bitter compound, because of the response with the bitter taste, the animal is going to drink less water. Okay? But now if we take a bitter taste cell, but we put a sweet receptor in it, okay? The animal, sorry, the opposite. We put a bitter receptor in a sweet cell, okay? So the beta receptor is going to activate that sweet cell and that sweet afferent, and the animal is going to keep on drinking, and it's going to drink even more because it thinks that it's drinking something that is sweet. So what this experiment tells us is that not the cell per se is encoding the information, but the afferent fibers and the connections in the central nervous systems are the ones encoding for what is sweet, what is bitter, and so on and so forth. Okay? Perturbations of the gustatory system can have profound effects on the overall head. Because beyond the hedonic values of consumption mediated largely by taste receptors on the tongue, recent work shows taste receptors broadly distributed in the stomach and throughout the gut. I mentioned this a few seconds ago. And these may contribute to nutrient sensing and glucose homeostasis and have been implicated in the development of type 2 diabetes. So a person having taste disorders can have a huge impact on the health overall of the patient. Taste helps us detect spoiled food or liquids. Usually those are bitter or sour taste. Some people can detect the presence of ingredients to which they are allergic to. Loss of taste may make people eat too little and lose weight, or eat too much and gain weight. And also, it can lead to patients adding too much sugar or too much salt in order to taste the food. And that can have a huge impact on developing diabetes or high blood pressure. The most common disorder on the taste disorder is phantom taste. Let's go again. Phantom taste perception. And it's a lingering of an unpleasant taste even though there is nothing in your mouth. The other disorders that we can find are hypogeusia, which is reduced ability to taste sweet, sour, bitter, salty, and umami. Ageusia, which is people cannot detect any tastes at all. Or dysgeusia, which is a condition in which a false, salty, or rancid or metallic taste persists in the mouth. And sometimes it could be accompanied, it could go with a burning mouth syndrome. True taste loss is rare, and most often what people are experiencing is a loss of sense of smell instead of loss of taste. Probably many, if not all of you, have a cold at some point. And when you have a cold, you eat because you have to, but not really because food tastes, okay? In reality, in those cases, what you lost is the sense of smell, not necessarily the sense of taste. But we say, like, food doesn't taste, okay? But we have to make a distinction. Both of them are very close, the sense of smell and sense of taste, okay? Usually, when they are combined, we talk about flavor. Flavor is the combination of the sense of taste and smell, okay? So the more accurate way of putting it is like food doesn't taste when you have a cold, it would be food doesn't have a flavor, okay? Because most likely you are tasting what you should. What are the causes of the gustatory disorders? Most likely infections of the upper or middle respiratory pathways. Radiation therapy for cancers of head and neck is very common on patients after thyroid cancer that go radiotherapy to lose the sense of smell. Exposure to certain chemicals, insecticides, medications, including common antibiotics and histamines. Head injuries. Some surgeries can affect because, some surgeries can affect ear, nose, or even the throat and affect the gustatory system. And poor oral hygiene and dental problems can also affect your gustatory system. And to finish with the gustatory system, here is how it looks like the fungiform papilla in a person that is a normal taster. Here is how it looks like in a super taster. So super tasters basically have more papilla than the rest of us, me in particular. Okay? Any questions about the gustatory system? Yes?
I've heard that there are some things that we can taste in food that's not placed in our tongue, we receive it through our skin. How does that fit into that?
We can taste things through our skin.
Yes.
I never heard of. If you can later on give me a reference or something, I can look into that. I never heard of that. Not at least in humans. No, because I think some type of frogs can do something like that, can detect things on the skin. Flies for sure. The taste receptors are on the legs, some of them. But in humans, I never heard of. So, we're going to move into the olfactory system. We know a lot of the olfactory system from, here is a representation from the 19th century of Ramón and Cajal, but there's a lot that we don't know about the olfactory system. So you're going to hear me saying a lot of time, I don't know. Here is a cartoon with a representation of a sagittal view of a human head. Here is a human mid-sagittal section. In the back of the nasal cavity, we have the olfactory sensory neurons. They cross the cribriform plate, which is at the base of the skull, into the olfactory bulb. And here is a ventral view of a human's brain. Here are the olfactory bulbs and the olfactory tract behind. So in this case, we remove the cribriform plate, so you're looking at the base of the human brain. So if I were to present you with specific wavelengths and or a color, most of you would be able to say like, oh, that's yellow. Or I can present you with an amplitude wave for a sound or a sound per se, and you would be able to tell me, oh, that's a high pitch or a low bass. And the same is true for the gustatory system. If I show you a molecule of sugar, or if I give you sugar, you're going to say like, oh, that tastes sweet. What about the olfactory system? If I present you with an odor, you are going to be able to tell me what it is. But if I present you with a molecule, there's nobody, and I bet there is nobody, in the room that can tell me what either one of them smells like. But all of them smell like something. Okay? So that is one of the problems that we face when trying to understand the olfactory system. The other problem that we face is that based on the structure of one compound, like this one, for example, it's really hard to predict how a very similar molecule is going to smell. The difference between these two is an oxygen or a carbon in this position. This molecule smells like ambergris. Ambergris is basically whale's vomit. Okay? So if you're walking on the beach and you see whale vomit, what are you going to do? Turn around. Wrong. Grab it. Perfume companies pay a lot of money for that because it's a main compound on the perfumery industry. Here, I mean, you can kind of like guess the shoulder of a person, the arm, so you can imagine the size of that. I think that piece was like $200,000. So next time you're walking on the beach on a romantic walk and you see that, stop the romantic part and grab it. So that is what this molecule smells like. What about this one? It doesn't smell at all. It doesn't have any odor. So here we have exactly the same molecule. Basically, they are optical isomers. This part of the molecule is over the ring, and this one is under. This one smells like caraway. This one smells like spearmint. So basically, from the structure, it's really hard to predict what it's going to smell like. Not only that, the same molecule at a low concentration can smell like floral. At a high concentration, it can smell like putrid. The olfactory or odors have another problem. It's that they move in 3D, basically. So here is a representation with two different colors, and it's small, but they represent basically an odor, and how the odor distributes in the air. And I think it's a bee or something, an insect in here. If an insect has to find the source of that molecule, or if a dog is hunting and trying to find a prey, or if you get a human to do it, you cover eyes, ears, and you put a special device to find a path along the way, we are all going to behave the same way. We are going to start doing this path that goes back and forth. Why? Because if this fly is trying to find the green source, it would smell, detect it, and all of a sudden it's gone. So it comes back, detects it, and it's gone. It comes back, and so on and so forth. Others moving what is called plumes. Okay? So that's why you see an animal moving like this when you're trying to find the food. So there are many things that we don't understand about the olfactory system. What about the ones that we do? Here is a representation of the olfactory system, of the olfactory epithelium that is in the back of your nose. What we have in the olfactory system are olfactory sensory neurons. These are the neurons that are going to detect odors. These olfactory sensory neurons are bipolar neurons. They extend an apical dendrite into the surface, and they have olfactory cilia that is in contact with the air. And in there, other molecules are going to bind. We also have supporting cells, as we saw in many other systems, and we also have stem cells that can regenerate olfactory sensory neurons. Olfactory sensory neurons in your nose are constantly dying. They are dying right now, and they are being replaced all the time. I'm going to use an example. At the beginning of the semester, in your noses, you have one set of olfactory sensory neurons. By the end of the semester, all of those are going to be gone and replaced. Nonetheless, the smell of coffee in the morning is still great. How does it happen? We don't know. This is a high magnification of how the cilia and the sensory neuron looks like in the surface. This is, basically here is the cavity, the nasal cavity in your nose. Here is the end of the neuron, the dendritic knob, and here are the cilia extending on the surface. The cilia, basically, what it does is increases the surface in order for us to be able to detect odors. And I'm going to turn a little bit to mice because it's what we know the most, but it applies to, most of this applies to humans. Here we have a rodent head side view. Here is the nasal cavity, the nasal epithelium, and the olfactory bulb where it projects to. So here are the cells that I just talked about. The olfactory sensory neurons with the cilia in the nasal cavity. I mentioned before axons that cross the cribriform plate and they target the olfactory bulb. Okay? So, in the cilia, we have the sensory receptors, the olfactory receptors that are going to detect the odor. These receptors are GPCRs, G-coupled protein receptors, and here is a representation of one of them. The other molecule binds, and the same that I described for the gustatory cells applies here. A G protein is activated, secondary messengers are activated, and cyclic nucleotide gated channels open, calcium enters, potassium exits the cell, and an action potential, if the stimulus is enough, an action potential is triggered. Okay, and it travels in the axon through the axon into the olfactory bulb. Remember, olfactory sensory neurons and gustatory cells are the only two sensory cells that we talk about that can have action potentials. We talk about 5 receptors for gustatory cells. Bitter, sweet, sour, umami, and which one I'm forgetting? Salty. 3 receptors for the visual system, cones, rods, and 1 for each wavelength. How many receptors do you think we have in the, for olfaction? Any guess? So, if we look at mice, rats, dogs, or possums, they are close to 1,000 different genes for detecting odors. If you look at humans, chimpanzee, or chickens, the number is lower, but still, we are in between 300 and 400 receptors. To put this in perspective, 1.2 to 4% of the entire genome is just odorant receptors. Okay? So how does the system work? We have tons of olfactory receptors. So each olfactory receptor detects one odor. Now, if we take this one, for example, it's going to respond to many molecules. Okay? So maybe the opposite. One molecule binds to only one receptor. Wrong again. One given molecule can respond or can bind to different receptors. So it's not as straightforward as any of the other systems that we talked about before. What we do know is that each olfactory sensory neuron that we have in our nose expresses only one odorant receptor. So basically, one sensory neuron pick up one gene out of 1,200 in mice or about 400 in humans. Only one of those. So if I go back to this schematic that I showed you before, in reality what we have is something like this, where each sensory neuron, each color represents a cell expressing one odorant receptor in particular. And although they are all mixed up in the epithelium, as the axon travels, crosses the cribriform plate and gets into the olfactory bulb, all the axons expressing the same odorant receptor converge into one structure called glomerulus, and inside any given glomerulus, all the axons express the same olfactory receptor. So basically, what I'm going to is like, it seems there is a map in there, okay? Because all the sensory neurons expressing the same odorant receptor go exactly to the same place, okay? That is also true for humans. I mean, again, I'm using the mice as an example because we know more, but this is also true for humans. We have the olfactory sensory neurons expressing one specific odorant receptor, the axon that crosses the cribriform plate, and all the axons converge into one specific glomerulus. Okay? And if we look at the cross-section of a mouse brain or a human olfactory bulb, the structure is very similar in terms of like it has different layers. So what we are understanding from mice, it applies, most of them, it applies to humans also. So going back to this schematic, basically, if we mention that each sensory neuron expresses one OR, and all of them expressing the same OR goes to one place, basically what we have in the brain, in the olfactory bulb, is a map sort of like this, where each glomerulus is sensitive to a particular group of odorants, because only one odorant receptor is expressing that glomerulus. So again, I'm using the concept of that apparently there is a map. We have the olfactory sensory neurons, and the axon of these olfactory sensory neurons form the cranial nerve number 1 that crosses the cribriform plate, and they target in the olfactory bulb these structures called glomerulus. From there, from the olfactory bulb, these cells that are shown in here, that are called mitral cells, extend an axon that form the olfactory tract. And that olfactory tract targets all these structures: piriform cortex, olfactory tubercle, amygdala, and the entorhinal cortex. From the olfactory bulb targets, we have projections that go into the orbital frontal cortex, into the thalamus, into the hypothalamus, and also into the hippocampal formation. There's something I want to point out in here. You've seen that before for all the other sensory systems. Before it reaches the cortex, it has a relay in the thalamus. The olfactory system is the only sensory system that reaches the cortices before it has a relay in the thalamus. Okay? How is it possible to have all these targets first, and the map that I just described in the olfactory bulb, how it translates into the olfactory bulb targets? Going back to this schematic, here we have the mitral cells, and each mitral cell is color-coded because the information they receive comes from one single glomerulus. So all the sensory neurons in there have express one odorant receptor. So this mitral cell is responding to one specific type of odorant receptor. This is how a mitral cell looks like. Here, the tuft shown in here in the apical dendrite, shown here. The secondary dendrites represented in here, and the axon shown in here. What I'm going to show you next is that individual single axon that shown in here, how it targets higher cortical areas. Sorry. And I'm going to use, again, a mouse brain. So I'm going to show you a projection of one individual mitral cell that is receiving a specific input from one set of sensory neurons that express one odorant receptor, how it targets the brain. That is the projection of that single individual axon that I just showed from one mitral cell. It targets basically everything. The anterior olfactory nucleus, the piriform cortex, the olfactory tubercle, the amygdala, the lateral and entorhinal cortex. Now imagine that by thousands of mitral cells in the OB. That map that we had in the olfactory bulb is gone. So there doesn't seem to be a map representation of the olfactory, or the other molecules in higher cortical areas. Okay, it's nothing like what we talk about for auditory, visual, or vestibular or gustatory system. The olfactory system, I mentioned, it targets a lot of areas. It targets basically what is called the paleo and the archicortex. And these are portions of the cerebral cortex that develops in association with the olfactory system. And in here, I listed all the structures associated with the paleocortex and all the excuse me, the structures associated with the archicortex. Okay? In red, I highlighted the ones that receive direct input from the olfactory bulb, and in blue, the ones that receive indirect input, meaning it's not straight from the olfactory bulb, but from the olfactory bulb targets. And there are two in particular that I want to highlight, the hippocampus and the amygdala. These, as you know, are heavily involved in emotion and memory. Okay? And it is believed, it's hard to prove, but it's believed that these projections from the olfactory bulb into areas that have to be with emotion and memories and that are with emotion and memory are the reason why, for example, we open a cupboard at home and we smell something and we remember our grandmother because it's the same smell that our grandmother had in her cupboard. Or we are walking by, somebody walks in front of us and the perfume that the person is using triggers the memory and we see the face of somebody that used to use that same perfume. Okay? The olfactory system seems to be very well associated with emotion and memory. What's the relevance of the olfactory system? It works as a warning signal. So we can detect there is a gas leak, smoke, a spoiled food, or dangerous fumes because of our olfactory system. Okay? And when the sensory smell is impaired, a lot of people change the eating habits. I mentioned that as when you're having a cold, food doesn't really taste, so you eat not that much, or you start adding more sugar or salt in order to compensate. Imagine what it is to be like that your entire life. So if you keep on adding sugar or you keep on adding salt, it could have a huge impact on overall your health. The loss of sense of smell can lead to depression. And there is so far anecdotal evidence, but it seems that before any patient has symptoms of Parkinson's, Alzheimer's disease, or MS, multiple sclerosis, they already had a huge deficit in the sense of smell. Although this is gradual, and patients usually don't report that. But patients that have Parkinson's, Alzheimer's, or multiple sclerosis, when tested, they showed that a reduced sense of smell. So there are a lot of studies trying to see if the sense of smell can be used as a predictor for some of these diseases. And also, it's important for neurogenesis. I'm going to get back to that in 2 slides. 1 to 2% of North Americans report problems with the sense of smell. The most common ones are hyposmia, which is a reduced ability to detect odors, anosmia, which is a completely lack of sense any odors. There are rare cases in which people are born without the sense of smell, and it's called congenital anosmia. Parosmia is the change in normal perception of odors, such as when we smell something that is familiar but is distorted. And phantosmia is the smelling of something that is not really there. Causes are all over the place. Aging is the most common one. With age, we lose the sense of smell. Sinus and upper respiratory infections is a big cause. Smoking, growth inside the nasal cavity, head injuries, or hormonal disturbances. Dental problems, also exposure to certain chemicals such as insecticides and solvents. People working in mines or in very dirty environments have the sense of smell affected. Medications, some antibiotics and antihistamines. Again, radiations in the area of the neck for treatment of cancer. And as I mentioned before, conditions such as Parkinson and Alzheimer can affect the sense of smell. Luckily, the sense of smell recovers. There is what is called adult neurogenesis. I'm going to probably get back this later on the course when we talk about development. But in the olfactory system, we have 2 places where there are new neurons being born. One is in the periphery, inside your nasal cavity. The neurons in the olfactory epithelium are replaced constantly, as I mentioned. And also in the olfactory bulb, inside the brain, we have new neurons, granule cells, inhibitory neurons that are being generated in the subventricular zone, right on the wall of the lateral ventricles. There are new neurons being generated that migrate into the olfactory bulb. And there's interest in this because together with the hippocampus in the dentate gyrus are the only 2 places inside the brain that we are generating neurons, and there's a lot of interest in using them as for therapy. This is not really an olfactory system disorder. It's called rhinetotilexomania. Rhino means nose, tilexis, habitual picking, and mania is obsession. And everybody saw this sometime or one time or another. And finally, I'm going to finish with a video that is going to hopefully show you the really important of the olfactory system. And with that, if there are no further questions, no? Okay, we are done.